Okay, so good afternoon, ladies and gentlemen. My name is Marek Babusiak, and I will be your host and panelist today. I'd like to welcome you all at Symmetric's free webinar, Accelerating Gene and Cell Therapy Trials with Real-World Evidence. So this exclusive webinar will concentrate on the newest strategies to successfully design and execute gene and cell therapy trials using the real-world evidence. We will hold this webinar in two parts. So it will start with a presentation, and then it will be followed by a Q&A session. For submitting your questions, please type them using the Q&A button on your Zoom panel. And if you have any technical difficulties, please use the chat function to message me directly. Now, ladies and gentlemen, it's my pleasure to introduce our trainer today, Mr. David Schwicker from Switzerland, who is the CEO at ORPHA Strategy Consulting. David is a well-known expert for accelerating time to launch and patient access for orphan drugs and advanced medical therapies. He's an author of numerous peer-reviewed publications, and he's a regular speaker at big international meetings, as well as distinguished Symmetric trainer. Hello, David.
Thank you, Marek. Hello, good afternoon to you. Good morning to everyone.
The floor is yours.
And so I'd like to start with a short preamble of a topic which is very important to me personally, but is also key to the discussion that we're having here today, which is how do we accelerate our gene and cell therapy programs. Essentially, what we're trying, which is patient centricity, patient engagement. Meet Eleanor. Eleanor is a young girl who lives in Paris, France. This is from the Eurodis. This picture is from the Eurodis Photo Award, which is the umbrella organization of the different rare disease patient groups across Europe. They do a competition every year, rare children photographed by their parents. This photograph of Eleanor, who has Rett syndrome, which is a rare, rather rare disorder that affects mainly young girls. It is a monogenic disorder. Therefore, it represents a very good target for potentially a gene therapy. And I believe there is at least one development program from Avexis ongoing in Rett syndrome. And I'm just highlighting this as a single example, not in the least because the patient organization rettsyndrome.org has done some excellent work in the last 10 years. They have invested approximately close to $50 million into a natural history study of Rett syndrome, published dozens and dozens of original articles on the natural history of the disease, 3 of which I've just highlighted here as a selection. As you can see, Rett syndrome affects nearly every aspect of these young children's lives: awake breathing disturbances, behavioral, cognitive disorders, very often misdiagnosed with autism spectrum. Therefore, also diagnostic criteria, clarifying lessons from the natural history study. And the importance of a natural history study, specifically in the rare disease space, and why I'm leading off with rare diseases is obviously, yes, a very large proportion of the over 7,000 rare diseases that are known today are of genetic origin, and therefore they represent excellent targets for the gene and cell therapies that we're discussing here today. So there is going to be a very large overlap. There's not necessarily an overlap, but there is, as of today, a 80-90% overlap between rare and ultra-rare diseases and then gene and cell therapy development as we're seeing it. What does this do just from the perspective of our clinical development program? Of course, if we have such natural history information, excellent information, peer-reviewed, published, it will inform study designs, for example, the population inclusion-exclusion criteria, duration of a study based on disease progression, biomarkers, all important. It can help us in collaboration also with the patient organization to incorporate patient-relevant endpoints. We can also, in discussing with patients and patient advocates, address patients' needs in the research, incorporate their viewpoints into clinical study designs, make our studies more attractive to the patients so that they will voluntarily agree to participate in these studies, which will enhance and support our recruitment. In all essence, we can de-risk these programs significantly, especially in the rare disease space and the gene and cell therapy space, through collaboration with patients, organizations, and that's why I wanted to put this ahead. So coming to the actual core of the webinar, this was like now a bit of a concert where you're expecting to see the main star, but somebody else was starting and playing before that. But definitely patient centricity is, of course, central to accelerating our clinical trials, especially in the rare disease and gene and cell space. This quote from David Lennon, who is the president of Avexis, in which he is quoted in 2019, "Incredible clinical promise out of a lot of programs, potential cures. However, not every program will be ready to move to the commercial stage because of the significant requirements in manufacturing." And of course, manufacturing is a huge hurdle. We'll not be talking about this in the majority today. We'll be talking about the clinical challenges. But as we accelerate our clinical programs, it will be all the more important to keep a close eye on the manufacturing side as well, because you will have accordingly less time to move then from the clinical to the commercial manufacturing batches. And in fact, we've seen this the case of gene therapies being delayed in terms of their market access and commercialization. Basically, the clinical trials being completed, however, manufacturing not yet up to the necessary requirements, or simply also capacity. We've seen capacity limitations in CAR T cell therapy, for example, as well. So this is an important factor. In addition to clinical trials, we all know the probability of success of clinical trials. Overall, if you look at the right-hand side here of this slide, these are just the newest numbers that I found. Probability of success from phase 1 to approval, approximately 10% across all the different major therapeutic areas. However, in cell and gene therapy, as estimated by MIT New Dig's Focus project in 2018, only approximately half of that, 5%. Definitely then the motivation to de-risk these types of programs, a very high priority for us. Nonetheless, Focus estimates that by 2030, we're going to have about 40 to 60 launches of gene therapies and up to 500,000 patients treated. This now based on the US market, and then you can accordingly extrapolate to your area and the rest of the world. De-risking clinical development, one of the areas very clearly that we see the potential to de-risk is in choosing the correct indication from the onset or set of indications. Again, here the probability of success rises from phase 1 to approval, rises to 25.3%. So a significant increase if you look at the rare disease space specifically. And interestingly, this is very closely matched in programs with selection biomarkers. And this is not really a surprise because many rare diseases, as I've mentioned, are identified by specific genetic mutations. Therefore, we would expect the success rates to match about. So choosing the correct indication and then, of course, also having companion biomarkers within the clinical development program would be highly recommendable, especially if we're in the higher risk situation of cell and gene therapy. The next aspect is, of course, the cost of the trial. Here specifically, the cost of the pivotal trial. I spoke recently with a serial investor in the rare disease space, also moving into gene therapy investment, and this person told me that, normally I invest till proof of concept, so I do a phase 1 and a phase 2 or phase 1/2 trial, and then I out-license the development assets that I choose to invest in. However, I always try to invest in those assets where, if necessary, I could move towards doing, from a financial standpoint, from financing the phase 3 trial if I can't get the deal that I want at my proof of concept stage. So clearly also for an investor or for a small biotech, what does it cost to perform that pivotal trial in that indication that you're doing is going to be of key interest. This here is a publication from 2018. What these authors did is they looked at a total of 138 clinical trials. Again, FDA approvals of 59 new therapeutic agents. They ran the cost of these trials through a standard CRO costing software and came up with a median estimated cost of 19 million, although, of course, with a very large spread. As you can see here, certain trials costing well over 100 million to a few trials costing less than 5 million, which were trials without a control group, which were in 3 orphan drugs with less than 15 patients each. In fact, the single largest factor driving cost was the number of patients in the trials. Not surprising, of course. Accordingly, also the number of sites and countries that you have included in those trials. So looking at an indication and understanding how large that pivotal trial will be, is there a good feasibility that we can get with one single phase 1/2 pivotal trial to an early approval, accelerated approval, conditional marketing authorization at the EMA, is going to be an important factor in deciding where you invest. This is a summary and, of course, also a bit of a blue sky wish list of where you would invest, but it gives us a good idea of the different aspects that are crucial in terms of choosing and prioritizing indications. Often investors speak about the tractability, which is made up, obviously, and I've already mentioned this on the example of Rett syndrome, a good understanding of the natural history, pretty good scientific consensus on endpoints of a trial, ideally well-established biomarkers, validated biomarkers, monogenic disease. Early data, obviously very good preclinical data, a very specific and good fitting animal model. Again, it's an ideal wish list. As also, on the one hand, we'd like to have a monogenic disease. On the other hand, we'd like to have multiple disease targets, ideally even on one platform or in a single product, a whole pipeline. So perhaps half a dozen rare diseases. And we've seen success stories like this in the rare disease space in the past, where you start with a single ultra rare disease that in and of itself doesn't necessarily have a great commercial potential. But then as you move along, you add further indications, plus as all of a sudden there is a treatment in the area, additional attention by clinicians is focused on this area. And what was originally thought to be an ultra-rare disease with maybe only 20 or 30 cases in a given country turns out to have many, many more cases, which then are promptly diagnosed and can benefit from a novel treatment in this area. Already mentioned the well-established patient advocacy groups. If you look at the requirements for let's say breakthrough therapy, as well as being included in EMA's PRIME scheme, they have to be serious conditions, so highly debilitating and or life-threatening, high impact on quality of life, as well as high unmet need. Of course, no existing treatments or not satisfactory treatments currently on the market, so a very high unmet need in this area. Ideally, of course, you've identified patients, your patient population, you have excellent contacts to the very few centers of excellence in each of the countries, which altogether brings you to a relatively limited scope of your clinical program, looking at that first or maybe second ultra-rare or rare indication. Smaller trials mean faster trials, fewer patients, fewer delays in recruitment, fewer sites, and lower cost. So that all sounds pretty good, pretty logical. Now I'm going to throw a wrench into the system, which is the affordability. The affordability of gene therapies, cell therapies is a topic. A little bit less acutely in the United States, where with the different private insurance companies and let's say the segmented population, basically in some of these ultra rare cases, you have per insurer 1 or 2 potentially cases a year. So the absorption of, let's say, a 1.5, 2.5 million upfront cost for a treatment for 1, 2, 3 patients a year is more tractable than, let's say, if you are a single payer, such as in Europe, and you have the country and you have potentially 100, 200 cases in the entire country. And then also there are a number of challenges. Everybody is talking about value-based pricing with regard to gene therapy, for example. But we have very little consensus between the different stakeholders what that actually means when you get down to it. And then, of course, while the individual budget impact of a product, let's say a gene therapy for spinal muscular atrophy, is relatively limited, especially SMA type 1. The cumulative budget impact of 30, 40, 50 of these gene therapies reaching the market in the next 5 to 10 years is going to be significant, as well as the growth, of course, in this area. Part and parcel of this is, of course, that we have less mature data at launch because we're trying to compress clinical development programs, and we just don't know about the long-term effectiveness of safety. Do we have long-term issues? Are these therapies really somatic? Are they germline? Do they enter the genome of patients? Do these therapies have any other unknown long-term toxicities that we absolutely ignore at this point in time? Or what about the long-term efficacy, for example, of CAR T-cell therapy that then still requires patients to have hematopoietic stem cell transplantation further down the road? And we're seeing this actually in a number of cases now where you have a combination of CAR T-cell therapy as well as HSCT for patients to optimize outcomes in these severely sick patient populations, with payers, of course, saying, okay, I'm doubling up on very, very costly procedures here, rather than replacing one costly procedure with another. Part of this is the high upfront cost, and we still have no consensus on cost effectiveness and how do we evaluate a cure. Certainly, the forever and still popular QALY does not cover all the dimensions of benefit that we are looking for and all the dimensions of value for these complex products. So why accelerate these trials with patient-centric? And this is the important aspect: patient-centered real-world evidence. So if I'm speaking about real-world evidence, I always mean it's patient-centered. So centered to all our thinking and all the discussion here is always to reducing the number of participants in the trial, both those exposed to the experimental treatment, as well as those, and perhaps especially as well as those exposed to placebo or sham procedures. Ideally, we would even like to eliminate these placebo arms if possible, which basically also comes to us from the patients. The basic message that we receive from patients is just simply that they are no longer willing to enroll their children into trials where they would have to undergo sham procedures. We want to understand, of course, the disease progression. I've mentioned this a number of times in connection with the natural history. We would also use natural history to produce historical control data for comparison in single-arm trials. You can call them external controls, some call them synthetic controls. We would combine this with many-to-one matching methodology. So we compare one single study participant with several historical controls, increasing the power of this analysis and argument. We would be using real-world enrichment to define a narrow patient population to improve the probability of detecting a treatment effect, so increasing the power of the study with the same limited number of patients that we would enroll. And yes, the irony of this doesn't escape me, of course, which is that normally in real-world evidence research, we're trying to broaden the population and trying to argue that clinical trials are too specific and that with real-world evidence, we should have a broader look at the population actually treated in real-world practice. And here we're using real-world data, real-world evidence to actually do exactly the opposite, which is granted something that we would do to accelerate our clinical program, to have an early approval, and then move towards the broader population step by step as we are on that market with a conditional marketing authorization, and we continue to generate evidence once the product is on the market. Again, recall the argument for a conditional approval is that the benefit to public health outweighs of an immediate availability of such a product outweighs the risk of an undetected safety signal due to the immaturity of the data. This is what the regulators postulate. Of course, our health technology assessment and pricing and reimbursement agencies are not always of the same opinion, but that goes beyond the scope of this webinar. And of course, we would like to, with our patient-centered work, incorporate patient needs into the studies. We would like to reduce barriers to participation of patients, accelerate recruitment, because recruitment is the one thing that, especially in the rare disease space, holds up our trials. Half of trials do not meet their recruitment targets. So it's the one thing that we need to focus on specifically with patient engagement, with patient-centric data, so that we can create these trials in a fashion that patients will want to voluntarily enroll. Also with regard to our competitors, because patients, especially rare disease patients, are very, very well informed nowadays. They have excellent resources and within social media, they have excellent networks. They know what's coming along. If in terms of their disease progression, they might be able to afford it, they will potentially wait to enroll themselves or their children into the trial of your competitor that's coming along within 6 or 9 months from now, that might not be placebo-controlled or have a sham procedure included in it, and be more patient-friendly and address their needs better. Okay, again, I'm thinking that this audience mostly is well informed about real-world evidence. So I'm not going to go into the basics. Real-world evidence 101 webinars are available, many of them. What I'd like to say about real-world evidence is that the approach that we're taking here is that it's not a dichotomy. So real-world evidence does not replace the randomized clinical trial. The randomized clinical trial remains in this philosophy, in this strategy, it remains the standard. This here on this slide in the upper left-hand corner from the controlled setting and a clearly interventional trial. But that because we know of the limitations in the indications that we're in, we're using the maximal amount we're maximizing our toolbox. So we're using the maximal amount of methodologies and strategies that we can use to generate the evidence that we require and connect a totality of evidence that we will then use for our regulatory approval packages, as well as then further down the line for the value evidence and negotiating pricing and reimbursement. So this goes from the traditional RCT, randomized, double-blind, placebo-controlled, over pragmatic clinical trials that incorporate certain type of real-world elements in arms, to your single-arm trial in case a randomization and placebo control is either infeasible or unethical in practical, combined with these historical controls, a randomized registry trial, or standalone studies which are purely observational, such as the natural history studies that I've mentioned, a registry, post-authorization safety studies, all the way over to our non-experimental, so all these retrospective real-world data sources that we hear a lot about and that are extremely useful for documenting epidemiology, standards of care, burden of disease, cost of disease, etc., etc. So our claims, our electronic health records, and a multitude of other sources, however, which are less controlled and which are routinely collected for reasons other than primarily research. So we have always the issue with the quality, the latency, the interoperability of this data. But we've overcome these to a great degree, and we're seeing very, very good studies being done, for example, in claims, which the FDA is also having an initiative in this regard, and they're continually publishing their findings in terms of replicating randomized clinical trial results with claims and EHR data. So this is a continuum, and we're using the total toolbox to create the totality of the evidence for our clinical development plan. What does the FDA say to all of this? I don't want to go into full detail in this slide. Basically, the FDA, and this is the summary of a number of workshops that the FDA held at Duke University, as well as their numerous guidances that they've brought out, although the final formal real-world evidence guidance we're still awaiting. So basically, it's a set of 4 questions. Firstly, what is the context? What are you asking us to decide? Is it a new indication? Is it a label revision? Is it a safety? What are your data sources? Are your data sources of sufficient quality? Are your methodologies of sufficient rigor? And all that together within the context of what you're asking us to decide, is your real-world evidence fit for purpose? My personal interpretation of this, and in several programs that we've worked on, this is an open invitation to be creative, to come forward with good ideas, high-quality ideas, methodologically, scientifically rigorous ideas, good data quality, and FDA will entertain it. We've got several hints in our gene therapy guidance for rare diseases. This is brand new, actually, off the printing block, January 2020. These were draft guidances issued in 2018. They've now become permanent or full-on guidances. As you can see here, there is talk of, yes, the RCT is the standard. Intra-subject control, very interesting for us. Blinding if feasible, single-arm trials and historical controls. If you use them, natural history is absolutely crucial. And the invitation, the suggestion to make your first-in-human study, your phase 1/2 study, adequate and well-controlled to support an immediate application, BLA. Long-term follow-up, basically, as discussed in this guidance, also from January of this year, it is a registry. And in fact, I'm not aware, if someone is aware of a gene or cell therapy that is currently on the market that does not have a mandated long-term follow-up in the registry, please tell me so. This is absolutely a standard that we're seeing now. And that many, of course, investors in this area and developers tend to forget. A fast approval is one important milestone. However, you're going to be continuing generating evidence on your gene therapy for 15 to 20 years after it is on the market, within every treated patient, but then, of course, in additional indications as well. On the EMA side, we have this publication, very interesting, which is done by the pharmacovigilance and epidemiology departments at the EMA. It gives us a very nice overview of the rationale and the cases where EMA has used real-world evidence, real-world data in their regulatory decision-making thus far, so status 2018, specifically looking at the CAR T-cell therapies, Kymriah and Yescarta, long-term follow-up in registries, Zalmoxis, a cell therapy, historical controls, Strimvelis, if I remember correctly, historical controls and long-term follow-up, Spinraza, long-term follow-up. Again, with registries. So this also a publication that I recommend. Let's look at a few specific strategies and case examples. So here, this is a classical case example. Granted, it is not a gene therapy or cell therapy in this case. It is enzyme replacement therapy. However, it is really this classical case, which I think is a one-to-one transferable to the gene therapy space. Again, this is in CLN2, a form of Batten's disease. Again, a rare disease significantly impacting these young children. First off, doing a longitudinal natural history study, measuring the course of disease, age of onset, diagnosis, progression, a rate of decline in motor and language scores, leading to probable duration and endpoints for the trial, as well as historical controls. Then discussing very intently with the regulators, EMA as well as FDA, on the data coming from this natural history study, designing the pivotal trial, addressing specifically the methodological statistical concerns of many-to-one matching, using different covariables, using these historical controls as a control group, leading then to a single pivotal trial, single-arm pivotal trial, enabling this approval for Biomarin, both in the United States and Europe. So I think a classical case. Then we have the randomized delayed start trial. This is especially good for a condition where you have a relatively stable disease or where you don't have too fast progression of the disease, because it is a two-stage design. So in the first stage, patients are randomized. They receive either the experimental treatment or they receive a control, so a real-world standard of care. And obviously, ethically, while they're in the real-world standard of care and would experience severe progression, that wouldn't be that ethical. But if it's a relatively slowly progressing disease, that is a design that is feasible. Then at a certain set endpoint, patients that are on control then actually switch over to the new treatment. So when you're enrolling patients or children into your trial, you can promise the parents, you can promise the patients that at some point they will switch over. Everyone is receiving the experimental therapy, assuming this is an indication where there is absolutely no satisfactory treatment on the market. Patients will not be inclined to participate in the trial if they receive initially placebo and have no chance of going over into the treatment group. It can reduce the bias by a certain degree versus a straight ahead single arm design. It can maximize the assessment of multiple outcomes because you of course have intergroup and intra-patient comparisons, treatment and control. So you can do several comparisons. This is also known, for example, as a blind start trial. If you can double blind everything, so you could initiate therapy in a double blind fashion. In one arm, you initiate active therapy immediately. In the other arms, you initiate a phase of placebo control and plus standard of care. And then in a blinded fashion, move over to therapy at a certain time point, 3 months, 6 months, 12 months, whenever is appropriate for your design. Obviously, with an actual procedure and a lot of cell and gene therapies, that blind start isn't as feasible as, let's say, with an antisense oligonucleotide with another treatment that you could provide to patients. This is an example of a randomized delayed start trial. So this is the pivotal phase 3 trial for Luxturna. This is Spark's gene therapy for inherited retinal dystrophy, specifically mostly patients with Leber's congenital amaurosis. This is a condition that is slowly progressing or hardly progressing at all. So entirely ethical to have a 1-year trial where you have a control group that has no intervention, and that after 1 year then cross over into the intervention group. As you can see here, a 2-to-1 randomization. Very few patients, because LCA, Leber's congenital amaurosis, is already an ultra-rare condition. And here we're talking about LCA that is mediated through the REP65 gene. LCA, or inherited retinal dystrophies, are not a monogenic disease. There's more than a dozen or even more genes that are responsible for these conditions. So we're cutting very thin here. We're slicing really thin. We're slicing an ultra-rare condition even more thinly by, let's say, even a dozen genes, and we're addressing the specific needs of this population here with Spark's gene therapy, Luxturna. And in this case, of course, the sham procedure was deemed unethical due to the risks of the subretinal surgery, especially because you had pediatric patients, I believe 3 years and older, were eligible to enroll into this pivotal trial. So you have also a remarkable case, as we see in other CAR T-cell therapies as well. You've got a mix of children, young adults, and adults, often in these very rare populations, in a single trial, in your single pivotal trial. What's interesting in this case as well is that the primary endpoint was specifically developed as part of this development program. So the traditional endpoints, which would be FST and best corrected visual acuity, were not specific enough from FDA's perspective for this population. They then worked together with the sponsor as well as the patients and experts to develop this new endpoint, which is a multi-luminance mobility test, MLMT, which is basically a bit of a maze that patients navigate through with different lighting conditions provided. And then you look at their progress moving through this obstacle course or maze as they go along. So a novel endpoint created specifically for this type of trial. And we're seeing this endpoint, MLMT, now coming into subsequent trials with Leber's congenital amaurosis or with retinitis pigmentosa, for example, as well. So a very interesting development in this regard. I had mentioned patient centricity. There was a patient-reported outcome tool here, a specific low vision modified version of the, I think, NEI-VFQ stands for National Eye Institute Visual Function Questionnaire. So 25 questions, 5 of which are dedicated to driving, which this population definitely cannot do. Therefore, low vision modified, but still not validated in this population. They did not get, contrary to all the other clinical results in this trial, they didn't get great results on the VFQ-25. As you can see, it's also not mentioned here on this slide. This did not, by the way, detract from the success. Luxturna had very good market access success as well, both in the United States, but also in the more critical European markets. Luxturna did have quite good success in terms of their market access. So a testament to Spark and their licensing partner Novartis also in Europe for that success in this area. The next strategy that we're looking at, so randomized delayed start, randomized real-world enrichment, I've mentioned it. Basically what you're doing here is you're starting your natural history study, you're starting an observational study. You are, the duration of that is suitable to your disease progression. Then in stage 2, you randomize those patients who have met certain enrichment criteria, be that a defined outcome threshold or be that a biomarker, and then you randomly assign them to receive your experimental therapy or remain on the standard of care. Again, it's post and pre-treatment progression, difference between observed predicted outcome, etc. You have a multitude of analysis options here. If you integrate it into an existing registry, for example, a rare disease registry, this is known as a registry-based randomized controlled trial. And now you're telling me, oh, David, give me an example. And I will be the first to admit that the examples of this type of approach as registration trials, pivotal trials, are still very few and far between. I think it's a potential opportunity, which is why I've included here two publications for your consideration, which sing the praises of this approach. And certainly it is the case that if you have a very rare population and an existing registry where the majority of patients are already included, let's say a pan-European registry or a US-based registry, you would be very, very well advised, obviously, to utilize the patient population in that registry and do retrospective analysis of the patients in the registry or prospective analysis as is necessary, and then randomize and recruit your patients out of that registry. I think it just makes solid sense. Of course, with the scarcity of the patients that are available, of course, this decreases by a certain measure your control over the trial because you have to work together with whomever, the patient organization, academic groups that run this registry. Finally, to conclude, I would like to briefly talk about master protocols, not umbrella and basket trials, which we know from the oncology space, but more platform trials. So platform trials are basically ongoing clinical trials. There's a given infrastructure. There's a pooled standard of care group, a pooled placebo group. There's a single protocol, a single IND, which continues, which is then modified. So protocol adaptation for additional experimental arms as new treatments come onto the platform. This is an example in acute myelogenous leukemia. The IND in this case is held by the patient organization. They run the entire thing. And as you can see here, the spokes on the wheel, the clinical sites, pharmaceutical companies that have agreed to test their compounds, for example, also CAR T-cell therapies, etc. Within this master protocol, the FDA is obviously involved. There's a genomics provider involved. The CRO is involved, etc. So you have an entire infrastructure ready to go that you can jump on. And of course, the vision is here that this basically replaces your phase 1/2 trial, that you graduate from the platform to phase 3 registration trials. You've basically agreed on this with the FDA before you join, which should dramatically shorten your development timeline. Of course, this is going on for 10 years now in the oncology space, but we're moving from oncology to neurology. The Healy Center for ALS at Massachusetts General Hospital is just launching its first ALS platform trial, again in the rare disease, rare neurological condition. And they have the first 5 therapies that are ready to enter. They've, of course, been held up a wee bit by COVID-19, as many things have. But they're getting ready to start this platform trial as well. I'm not aware of the 5 different therapies that are included. I don't know if they've published this, but there are gene therapy approaches. I don't know if they've already advanced from the preclinical to the clinical stage. Perhaps someone here online knows more about it in ALS. But it is certainly a possibility that you would have gene and cell therapies on such a platform. Then there's also an exciting research pilot getting started at NCATS, which is the PAVE-GT platform vector gene therapy, where they're intending to develop 4 gene therapies for monogenic rare diseases at the same time. By the way, this is not just a clinical trials platform. This is an entire platform. So the same vector, the same manufacturer, the same purification process, as well as the same clinical protocol, preclinical protocols, etc. So this is a complete platform that they're trying to develop and look at. So definitely also an exciting, let's say, initiative to keep an eye on. Why, and this of course a picture of pre-COVID times. I think it'll be a while before we see a packed stadium again, such as this. Simpler times, but why this comparison of a clinical trial to a stadium? And this was actually, I heard this in the webinar where they introduced the ALS platform trial, and I thought it was quite telling. You know, imagine if you held the football match and you built the stadium, you held the match, and then you tore down the stadium. And for the next match, you built a new stadium, held the match, tore it down, and did that over and over again. It seems ludicrous. But in the randomized clinical trial world, that is actually precisely what we do. We're building clinical trial resources one by one. In the face of ever rarer and more fragmented patient groups, with patients who are no longer willing to participate in trials unless these trials really address their needs. Patients are aware of the fact that they have a choice and are choosing their trials. They're acutely aware of the fact that they might not be eligible for a subsequent trial if they choose to enroll in your trial. Half of the trials do not meet their recruitment targets, which is a key limitation. So why are we continuing to build these stadiums and then tearing them back down and not using these platform trials? I really think it is the future. Don't take it from me, take it from Janet Woodcock, the director of CEDAR at the FDA. I personally believe the clinical trial system is broken. Master protocols, trial platforms, clinical trial networks need to be a future, and we need to utilize healthcare data, speak real-world evidence when at all possible. So I will end with, how do you operationalize all of this? There is a very excellent survey that Deloitte does year on year, and they ask how biopharma companies are approaching the implementation of real-world evidence. 90% of respondents are saying they're investing in building capability across the entire product lifecycle. So that includes as early as preclinical, looking at your indications, trying to understand these indications, trying to understand the patients better in these indications in order to choose your target, as well as to post-authorization, using real-world evidence for market access, pricing and reimbursement negotiations, using real-world evidence for innovative contracting, so outcomes-based contracting, as well as indication expansion. So it goes the entire life cycle of the product. Interestingly, 70% of companies are building internal resources, so they are planning to conduct a greater proportion of studies internally. We've got a rapidly evolving data landscape, so we're seeing combined data sources now. So for example, clinical trial data, genomic data, claims data, electronic health data. Again, this philosophy of the totality of evidence that I mentioned to you before. And we're seeing strategic data partnerships, the most prominent of one of which is obviously Roche buying Foundation Medicine as well as Flatiron, but other partnerships as well. And in rare conditions, of course, partnerships with patient organizations, genetic testing initiatives, diagnostic initiatives, purpose-built real-world history studies. So that's where you start. And in terms of operationalizing patient engagement, this is a current announcement. So the Patient Engagement Open Forum starts this Thursday. It is an event that is normally held in Brussels that has gone virtual. I encourage you to investigate whether you or someone of your team should participate in these sessions. Registration is free. There's no travel necessary, so this is relatively easy. We've got great sessions this week. We are starting off with, for example, incorporating patient engagement into early stage clinical research, incorporating patient engagement, patient viewpoints into regulatory processes. So some great sessions coming up this week as a recommendation. And now I think we still have, Marek, I don't know how much time we have left and for the Q&A session, and whether we received already some good questions or some questions are coming in for us from that. And
time
I'd
to
like to
ask the
thank you.
question. So basically, we have some 30 participants on board. So the forum is open for your questions. Type them in the Q&A button at your Zoom application, and I will read them for you, David.
Okay. So I think there was one or the other question that have come in ahead of time. So maybe you would want to use one of those beforehand while the others are maybe coming in, questions.
Just a second. Yeah, we had one or two. There is one. So we answer live. David, you briefly touched on the affordability issue in gene therapies. Should a clinical development program also consider the payer's perspective and incorporate endpoints and evidence generation from the value and pricing perspectives?
I think that, thank you. That's, of course, an interesting question, because the faster our clinical development program progresses, we're coming to the market, and let's say we're successful in obtaining a conditional marketing authorization, then with this premature data, as we've seen, for example, in the oncology space over and over, but also for certain orphan drugs, especially in the European context, market access with a conditional approval can be rather challenging. So the more data that you have that are of specific interest from the payer perspective, and if possible, being able to incorporate from the payer perspective certain aspects into your clinical development program from the onset, even if it's only in that single phase 1-2 trial, or alternatively in the natural history study, where you would be looking at the socioeconomic burden of disease and various healthcare resource use, which you wouldn't perhaps want to burden into your phase 1-2 study. That could be definitely an option if you are running a natural history study, or if you're collaborating with a registry. Those unmet need, burden of disease, epidemiological research that you would have to do for a national cost effectiveness and pricing and reimbursement submission is something that I would definitely take into account very early in development. Yeah.
Excellent. Thank you, David. There is another open question. The importance of patient centricity is well understood. I'm wondering how well patients know about clinical trials and the development process, and how we concretely engage with patients as part of clinical research in actual practice. So it's really beneficial to both sides. Do you have concrete examples of companies doing this successfully?
Mm-hmm. Yeah, thanks. I think this is a very interesting and important question, which is why I actually mentioned the Patient Engagement Open Forum, and which starts this week. This, for example, was presented by Janssen at last year's Patient Engagement Open Forum, and what they call this is a patient engagement dashboard. I just put this into the supplemental files slides because I like this approach, which is they have a number of questions that they ask themselves throughout their early clinical development program. Yeah. So in terms of the strategy, do we have a patient journey complete? Do we have direct patient input into our disease area strategy in terms of selecting our indications where we're going to invest and develop in? What about the target product profile? Do we have patient perspectives and insights from patients directly incorporated into our TPP? What about the endpoints? Do we have patient relevant endpoints You know, do we have a pre-approval patient access strategy? We have not spoken about early access for patients here as well from different rare disease development programs. We know that we could collect data in expanded access and early access programs as well. And this is data that we can use as supplemental evidence and supplemental information for our submissions as well. Did patients provide input into the clinical development plan? Our protocol's designs and operational strategies informed by direct patient insights. So are we asking patients to come in to non-virtual visits? You know, in a post-COVID world, how many absolutely non-virtual visits do you still want to have in that trial? Do you want to obligate patients to come into 2 screening visits plus 14 site visits in a single year? You know, do we have PROs included? So I think this is one example where I would say you can use these questions and pose them to yourself at pertinent time points in your development program and ask yourself how you have actually done this. The other question that is related to this is, you know, how informed are patients about the clinical trial process, about regulatory processes, etc. And the answer to this, of course, more and more. One of the co-sponsors of the Patient Engagement Open Forum this week is UPATI. UPATI is the European, what should I say, patient university that specifically has the task of you know, educating patient experts on the clinical trial process, on regulatory processes, on health technology assessment processes. So you have more and more clinical trial literate patients that you can consult as experts, that you can bring in person or virtually, and have a seat at the table, and that are literate in these things and can contribute to the discussion. Now, these might be patient advocates that don't have the detail of the actual indication, especially if it's a very rare and special indication that you're looking at. And so you might have a mix of these, you know, highly educated patient advocates, as well as specific patients that can go more into the depth of, let's say, the patient experience, the lived experience with the indication.
Yeah.
Okay.
Perfect. Thank you for this in-depth answer. We have another question from our attendee, Matej Boda. Regarding platform trials, I'm interested in possible issues of assuring scientific integrity, including data quality, confidentiality, for example, in pharmacovigilance, particularly if we, as the sponsor company, don't hold the IND. I mean, this is experiences have been made in practice with platform trials in this regard.
I think that this is a core question. This is a core question because, you know, it's not only a standard operating procedure, it's nearly hardwired in the biopharma industry that we hold our cards very tightly to our chest. We want to control all aspects of that trial, which is why we prefer to build the stadium, run a match, and then tear it back down, because we want to control every single aspect of the trial. We want to control the data. We want to control the quality, etc., etc. So all of these strategies, pre-competitive data collaborations in real-world evidence, pooled placebo control groups, a pooled natural history registry, other pooled registry initiatives, such as post-authorization registries from the CAR-T side, they include letting go by a certain degree of the control that we're used to. And this is a very important philosophical step that is not going to be easy. It's also not going to be easy to sell that internally to the decision makers at the companies. But I think as we see it becoming more and more difficult to run these standalone clinical trials, I think we will see these platform trials emerge more and more. Case in point, the COVID-19 health crisis, where all of a sudden companies are super willing to jump on to trial platforms, master protocols, discussing with the FDA, especially in the treatment space, a little bit less in the vaccine space, but they're also trying to pool resources to move as quickly as possible. I think this is a great example, and we're always talking about how COVID-19 is going to push the digitalization and virtualization of trials in the future, something we haven't spoken about here today, but that is certainly a topic as well in rare diseases. But I think also COVID-19 crisis is going to give a push to these master protocols and just, moving together for speed. But it takes a different mindset, that's for sure.
Yeah. Okay, so waiting for more questions. The Q&A pool is open. No more questions so far, so I guess
I think that's fine. I mean, I don't know if we scheduled for an hour or not. We're a little bit over the hour right now. So maybe, So
let me thank you, David, again for excellent presentation and very nice answers to the questions we obtained. And thank you all attendees who joined us today at our Symmetric webinar, Accelerating the Gene and Cell Therapy using the real-world evidence. It was great to have you. So we would like to pay attention at our proper training, which will be in November in Vienna. And this one will be dedicated to the development of advanced therapy medicinal product in more detail, including the real-world evidence and patient centricity and all the.
There will be some part of the commercial side as well, so market access, pricing and reimbursement in brief as well, some business planning, investment planning as well. So pretty broad program, but also available as online training. As
so you can either participate in Vienna or you can tune in and turn on.
So thank you everyone for attending today. We will make the slides. We hadn't said that, should have said that at the beginning, sorry. Of course, we will make the slides available to everyone, and we're also going to make the recording available in the coming weeks. So thank you very much.
Thank you very much. Have a good afternoon. Bye-bye.
Bye-bye.